My name is Barrett Rollins. I was the Chief Scientific Officer here at Dana-Farber. I did that for about 16 years, but I've been at Dana-Farber since 1983. I went to medical school thinking that I would be interested in cancer. I always thought that that was the most interesting area of medicine to work in. I got interested in research as well as clinical care. And once I realized that that's what I wanted to do, a mixture of clinical care and research in cancer, there was really only one place that I thought of coming, and that was Dana-Farber. And so I tried to do everything I could to arrange my sort of burgeoning career so that I'd have a chance to come here. So when I first walked into the doors, I was an intern. That was in 1980. I was lucky enough to be a fellow here in 1983. The sort of tenor of cancer research and cancer care at the time was very different from what it is now. It was very empirical. There were drugs that seemed to work in some cancers and didn't work in others. There were some cancers that were utterly intractable to all of the sort of interventions people were trying. And I think that a lot of people in internal medicine, when they looked at this subspecialty, kind of threw up their hands and said, why would anybody want to do this? Of course, this was the biggest need in medicine at the time, so it was an exciting thing to get into. Because things were kind of empirical and kind of small, I remember when I first came to Dana-Farber, the place was small. I think a lot of people refer to it in those days as a sort of mom and pop shop, and it was. Everybody knew each other. It didn't matter what you were doing or where you were. There was this sort of casual and very friendly attitude. I remember one of the things that encapsulated that best is that the person who ran medical records always brought her little dog in, and the dog stayed in its little bed next to her desk, and if you wanted to go down and get somebody's chart, you said hello to Mrs. Votter's dog. And that was the kind of place it was. That doesn't scale. And so when it became clear that Dana-Farber offered to the community in Boston and New England the opportunity to be the primary place to take care of patients' cancers, it had to enlarge. I would say that even though the sort of casualness didn't scale, what did scale was the sort of intimate feeling. There is a way that Dana-Farber still feels like a mom and pop shop. When I talk to my friends at other major cancer centers around the country, places that we have tremendous respect for, the feeling is quite different. And so somehow that feeling has remained, and that's been a core attribute of Dana-Farber Cancer Institute. So during my time here, what I think has changed most profoundly at Dana-Farber has been the way we are positioned in the community. It's not that the community didn't know about Dana-Farber or didn't respect it, it certainly did. But when I started here in the 1980s, the Institute was really focused on how to do cutting-edge research and cutting-edge clinical research in really difficult cancers. And because of that, there was a sense in which you might have considered the Dana-Farber to be sort of a boutique-y kind of place. What changed most profoundly, I think, was under Ed Benz's leadership, where his vision was that Dana-Farber should be bringing its advances in patient care to the community. We should be treating as many cancer patients in Boston and New England, around the country, eventually around the world, as we possibly could, because we had the most to offer. And what I've seen is this sort of switch. It's not a switch. It's an additional sort of part of Dana-Farber's mission to be able to accommodate the ability to treat all of those patients. What is important about that is that that occurred at the same time that our treatment options expanded. And so at the same time that we made that decision, what has changed most profoundly in cancer treatment is the availability of targeted therapies, immunotherapies. When I started as a fellow at Dana-Farber, if a patient came into my clinic who had metastatic non-small cell lung cancer that had spread out of the chest, you could pretty much start a timer. And 6 months later, that patient would be dead, no matter what we did. Chemotherapy, radiation, it didn't matter. Now, with targeted therapy alone, you see patients living 2, 3, 5 years. If one therapy doesn't work, you switch to something else. And then with immunotherapy, it's possible that some of these patients are actually being cured. That has profoundly changed the practice of oncology, and that's been exciting to be part of. My role at the Institute has changed really profoundly in the time I've been here. That might be true for anybody who's stuck around one place for 40 years, but even if that's not true, it's been true for me. So, I came here as a fellow. I was very lucky to be able to land a faculty job here. At the time, I was taking care of patients, but I also wanted to start doing my research. About 7 or 8 years in, I realized I'm just not good enough to do 3 or 4 things all at once and be really good at all of them. So I had to make a tough decision, and the tough decision I made was to give up clinical care and focus on my research. So I became a translational researcher, always with this focus on what I was doing in the lab that could affect cancer patients. But after 9 or 10 years of that, another opportunity came along to become the Institute's first chief scientific officer. That was an unexpected opportunity, something that I had to be cajoled to do, but it turned out to be the best job I've ever had. It was 16 years of really exciting work, and so that changed. And then after a while, it's after 16 years, it's time for somebody else to do that job, and now I've transitioned into something else. So for me, Dana-Farber has been a place where I've been allowed to do a wide variety of things that fit what I wanted to do at that time, fit my skills. It's just been a tremendous opportunity, and I hope that I've been able to help the institution as I've done all of that. So when I was chief scientific officer, there were a lot of challenges, a lot of opportunities. The position was created by Ed Benz with the advice of Stan Korsmeyer, because up to that time, you know how decisions at large institutions are made around very small tables with just a couple people? There was never someone from the research enterprise at those small tables until about 2004 when Ed created this job and offered it to me. And so I got to see firsthand how decisions were being made about what kinds of resources could be brought to bear around scientific problems that had to do with cancer. For all of that time, the 2 big problems were always space and money. And if you go to any institution, anywhere in the world that tries to do research, the problems are always space and money. But within those constraints, you find ways to do things that are really kind of interesting. I'll tell you about a couple of them. One was creating an animal imaging facility. We were a little bit behind when I started my job in being able to do longitudinal imaging of animals. In other words, to take the same animal, give that animal an experimental compound to treat its cancer, and then do what you would do with patients, although in a compressed time scale, do MRIs or CT scans over a period of time to see if the tumors would go away. That requires a lot of infrastructure. And what I came to understand was that the Longwood medical area is the most overbuilt medical area in the world. You couldn't find an extra 1,000 square feet of space to do this, much less the 30,000 we'd really need, plus all of the infrastructure to do MRIs and so on and so forth. And so that led to some really interesting sort of new things that I had not been trained to do, like dealing with city government, meeting Mayor Menino, meeting some of the city councilors in South Boston, and realizing that there was this incredible opportunity in what has since grown up to be the Innovation District for us to be able to plant our flag, to get a large amount of really high-quality space for very little money at the time. That's changed now, but at the time, very little money. It was at a distance from Dana-Farber, and that continues to be a challenge, but it was the only way we could do this. And in fact, through a lot of the work that we did, we were able to get $10 million towards the capital construction of this from the Mass Life Sciences Center. We found donors who were excited about this prospect. And when you put it all together, we have this amazing facility that allows us to do preclinical imaging, but it also has our cyclotron and our ability to make radiopharmaceuticals. And that was a very gratifying thing to be able to put together. So that's all about infrastructure. But the other really gratifying thing was around science itself. In about 2010 or so, a friend of mine, a colleague who had gone to Novartis, called me and said, "How come you can't tell me who all your patients are, who might have mutation X so that we can test our drug against mutation X in those patients. How come? Ordinarily, when I get calls like that from my colleagues in the pharmaceutical industry, I just kind of laugh and ignore it. But this one really stuck. Why not? And so that ended up being the Profile Project. It was one of the first projects in the world that did genomic testing on every patient who came to a cancer center. That was a really astounding project because it required the cooperation of somewhere north of 50 or 60 people, both from Dana-Farber and the Brigham, to be able to put together the process flows, the testing, the reporting, figuring out how we're going to get paid for all of this, put that all in place to be able to create a profile, which by now has become a standard part of our clinical interactions. And also, now that other institutions had started doing the same sort of thing, we combine all of our data into something called Project Genie, something sponsored by the American Association for Cancer Research. And we get to be one of the founding members of that because we were able to do the Profile Project. So that was also incredibly satisfying. So how do we decide where to put our resources in various different scientific opportunities? Because there are always a lot of scientific opportunities. There was a misunderstanding about my job. If you think about what a chief scientific officer does at a company, the chief scientific officer sets the scientific agenda, and the rest of the company follows that scientific agenda. If I, in my position, were to set the scientific agenda for Dana-Farber, my head would be on a spike on Brookline Avenue. My colleagues would not stand for that. So instead of that, what I tried to do in my time as chief scientific officer was to meet with my scientific colleagues as often as possible, listen to what they had to say, listen to their justifications for requests for resources, and balance those against other requests, and try in a sort of consensus-building way to devote resources where we thought they would make the most impact, where we could leverage the skill and brilliance of our scientists the most. I'm not sure that we always made the right decision, but I would venture to say that more often than not we did, which is why things have been able to advance. So I'll give you an example of where we did devote resources, where things have really paid off. And that was in chemistry. So the initial idea about chemistry came from Stan Korsmeyer, who unfortunately did not live to see the flowering of that idea. But around the time that I became chief scientific officer, Stan was still alive but died a few years later, we saw this as a major opportunity. This is one of those areas where every thread that I pulled on in the Institute told me that there was something good about chemistry, whether it was the clinicians, the basic scientists who wanted tool compounds to investigate biology, the pharmacologists, the cancer biologists, it didn't matter. People wanted our scientists to be able to make compounds that were well-defined and would do well-defined things. And so we doubled down on that investment, doubled down, tripled down. We devoted resources to recruiting faculty who were expert chemists, who could have gone to companies to become medicinal chemists, or could have gone to basic science departments in universities to do chemistry, but decided to come here because we gave them resources and they were able to see how their chemistry could impact the development of drugs that would actually impact patients. The other thing we offered in that domain was the Longwood Center. In the Longwood Center, we created first-class chemistry facilities so that people could do synthetic chemistry and make new compounds. And that then, in turn, led to a whole variety of interactions with industry. And that's where the sort of financial payoff of this came, that we start to bring money back into the Institute. That's sort of a return on investment that goes beyond the intellectual return on investment. In the old days, before we could actually do chemistry here, or at least before we did the kind of chemistry that companies would be interested in, we had biologists who would identify a target. They would use their cell lines or their animals or their genetics to say, well, here's a molecule we really ought to think about inhibiting because this ought to improve the lives of cancer patients, although we don't know for sure yet. We've always been an entrepreneurial place, always. And so we would try to get patents on those targets and then license those kinds of patents to industry. Well, Target patents, they're something, but they're not very valuable because all the risk in developing a drug comes from making the compound, testing the compound, seeing if it has toxicities. We weren't taking any of that risk. All we did was say, hey, look, there's something that might be an interesting target for a drug. We license it to a company, and then they do all this stuff to risk things. And so the deals that we could make, the licensing deals we could make, were not all that favorable to Dana-Farber. We'd get very, very low percentage royalties, for example, or we wouldn't get upfront payments for the licensing. But once we started making our own compounds and could demonstrate that they actually interacted with the target they were supposed to interact with, and we could go even farther because we had the animal facility. We could show that those compounds, in hitting their targets, made cancers in animals decrease, shrink. Now we had something where we have started to de-risk that property. And the more that we could de-risk the property inside Dana-Farber, the more we could ask for in terms of our licensing revenue. And so, while we bring more money into Dana-Farber, drug companies are actually happy about that. They're happy to pay that extra premium because a lot of the things have been de-risked already. And so I would say that our interactions with industry now are really quite different from what they were certainly in the 80s and the 90s, even in the aughts, because we are now much more of a partner in actual drug development. And we have new relationships with a variety of actors that allow us to push that even farther. And so that's been a monumental change in the way we do drug discovery at Dana-Farber. If I were to point to a couple of the things that I did that I'm most proud of, I think Profile would have to be at the top. That was really, the ability to do genetic analysis on every patient who walks through the door. That was a heavy lift. Nobody was opposed to it, but everybody had a reason why it wouldn't work. And to get everybody on board and moving in the right direction together was really incredibly gratifying. That was pretty wonderful. And I'd say the other thing that I'm really proud of is a little bit softer, a little more ineffable, which is that I think the scientists at Dana-Farber feel listened to, they feel heard, they feel like they are essential players. Nobody would have ever said they weren't, but sometimes if people don't feel like they're heard, they might not feel that way. And so I think the morale around the scientific enterprise at Dana-Farber is high, and I hope that I was able to contribute to that morale. Dana-Farber is the kind of place where we place a huge premium on mentorship. Everybody moves ahead because somebody has helped them here. And if I think about my career, there were at least 3 people who were really critical in terms of mentoring me. First is Chuck Stiles. Chuck was a professor in our Department of Cancer Biology, and I did my postdoctoral training with Chuck. Chuck was the person who taught me how to be a scientist, taught me how to think critically, taught me how to evaluate experiments, taught me how to take the knowledge that I had as a physician and use that to sort of color the experiments that I would do in his lab. Those are just invaluable lessons. The second person who really helped me was David Nathan. Now, David has helped literally hundreds of people. He was the person who convinced me to take the chief scientific officer job. I was resistant. I thought that the only scientists who went into administration were failed scientists, and so why would I want to do that? David was the one who sat me down and convinced me that this job, in fact, matched my skills, such as they were. And I started thinking about what his advice was, and I looked around and saw that he had placed people in these incredible positions around the world, and they were all happy with where they were, where they'd ended up. And it was because David told them what he was telling me. And I finally realized I would be an idiot not to listen to him. So he was just critical in terms of the development of my career. And then the last person who really helped me tremendously was Ed Benz. Working with him through most of my time as chief scientific officer was an unbelievable hands-on education, not just in how to be a good administrator, not just how to think about how to lead an institution, but how you get people to come along with you. For better or worse, I never had a budget as chief scientific officer. I couldn't use the best tool in the world, money, to get people to do what I'd like them to do. I could use moral suasion or whatever other tools Ed Benz taught me to use to get people to come along. So that was an incredible education. And those three people really have made my life at Dana-Farber what it is. You know, I have, after 40 years, a number of memories at Dana-Farber that are just sort of key. But in fact, what has influenced me most are sort of memories that trickle down through many, many years. So for example, I was fortunate enough to work here with my wife, Jane Weeks, for a long time before she died. We'd have lunch together, we'd come in together, we'd leave together. To be able to have somebody here that I could talk to about anything that was going on was really wonderful. And by the way, one of the specific best memories I have is that Jane and I both served on the committee that designed the Yawkey Center and the Yawkey Building. And that was really one of the most fun times to think about how to put together a building that would be, clinical, and listening to patients and patients' families about how, what would be best, and thinking about architecture. That was really a great memory. But the other sort of memory, if I could use the term loosely, has to do with the people that I interacted with here. My colleagues are unbelievable. And most of these people have turned into good friends. I mean, these are now my friends. This is sort of the family aspect of Dana-Farber. And if I think back about what I've gained over the past 40 years and memories that make me happy, it's dealing with my colleagues who are now my friends. So one of the things I started doing in the late 90s was volunteering with the Dana-Farber Marathon Challenge. Like most people, I entered it selfishly. I wanted to run the Boston Marathon, but I'm not an athlete. And there's just no way. You could put a gun to my head and you could chase me with rabid dogs and I would never be able to do a qualifying time. But if I raised money for Dana-Farber, I could run the marathon. So, it's a little transactional, but that's where I started. Well, my God, it was a life-changing sort of experience. And it was really life-changing in two ways. One was the people who work for the Marathon Challenge. As you know, they're community volunteers. These are people who are cancer survivors. They're relatives of patients who had cancer. Some are ex-faculty members. These are amazing people. I have loved working with them over the years. They're inspiring. And so that really made me think that this is more than this transactional thing that I had brought to it. And they really, I would do anything for these people. And then the other really impressive thing is to actually be out there in the course. So I've run for DFMC 10 times. I have obsessive compulsive disorder, so I could either run 10 times or 20 times. 11 was not, in the card. So I ran 10 times and I stopped. But each time, to be out there on the course and hear people say, "Thank you, Dana-Farber," is actually "Thank you, Dana-Farber." But to hear, "Thank you, thank you, thank you, you saved my life, you saved my child's life, you saved my parents' life." There's just nothing like that in the world. So that was truly an amazing experience, and continues to be an amazing experience for everyone who's involved. So I think Dana-Farber is in a great place right now. It needs to be in that place over the next 10 to 20 years. I don't think we're at the end of the sort of exponential phase of discovery science. I think that we're at this phase where the more you put in to the work, the more you get out. Chuck Stiles used to say that you're in a linear phase now. And so Dana-Farber has to keep doing that because we are nowhere near, I shouldn't say nowhere near, we are closer than we ever were before to helping every patient with cancer, but we could do so much more, and we can't stop now. And so I see continued science, continued clinical care being essential. Dana-Farber, after all these years, is still the only place I've seen that puts an equal emphasis on clinical care and cutting-edge research. We have to continue to do that to make the advances that we're going to need over the next couple of decades.